Hi, everyone, and welcome to our final installment of the Closer Look Journal Club for the fall 2022 semester. Thanks for everyone who's attended over the course of the semester, and thanks for those who are on the call today. We are delighted to be hosting Dr. Stephanie Lindsey from UCSD. She's a new assistant professor in the Department of Mechanical and Aerospace Engineering, started, as we just found out, summer 2021. A little bit of background about Dr. Lindsey. She originally got her Bachelor's of Science from Washington University in St. Louis, where I currently am at. And I mentioned that because she worked in the lab with Larry Tabor while she was here. And after that, she went on to do her PhD at Cornell with Jonathan Butcher. And she continued on to do her postdoc in the lab of Alison Marsden. She has a diverse background in terms of sort of bridging these multi-scale phenomena in the context of pediatric cardiac disorders. And in particular, we're going to hear about today some fluid dynamics based approaches, which is a sort of new topic for the Journal Club. Oftentimes we're focused on solid mechanics rather than fluid mechanics. So with that, I'll cede the floor to Dr. Lindsey, and we're happy to have you here today.
Thank you. Yeah, my experience as an undergrad at WashU shaped my whole research career, and so that's definitely why I do what I do. And that's actually when I first started working with the chick embryo in the Tabor lab, and that's still the animal model that we work with. So in my lab, we either do patient-specific simulations, with humans, or we do our own experiments using this chick animal model. And so that's what we'll talk about today, is chick animal model. Yeah, so I'll get started. So I have a bunch of motivation here. So our main motivation is that flow is a driving force throughout development. Shown here on your far left is a normal heart. Right in the middle, you have one of the major congenital heart defects. This is transposition of the great arteries. So your great arteries are your aorta, your pulmonary artery, and all of the derivatives. So all the flow that's coming to and from your heart is going to go through these great vessels. And so the precursors of these are the frontal arch arteries. So that's what we'll be focusing on. And shown all the way over here on the right is a single ventricle model. So one of the ventricles is severely underdeveloped. In this case, it's the left ventricle. And this is accompanied by this extremely narrow aorta. And so this is really characteristic. This narrowing in the aorta is really characteristic of this single ventricle defect. And so for us, we really want to know, what is the role of hemodynamics in all of this? And so this is just a postmortem section of the aorta, so you can really see this narrowing here. And so congenital heart defects account for over 29% of deaths from developmental abnormalities and 1% of infant mortalities. The heart's the first organ to develop because a blood supply is needed in order for everything to grow and live. And in the US, the majority of these defects arise from malformation of the outflow tract, which is these great vessels. And then you'll see how that, I'll show you how that evolves throughout development. So there's a lot of evidence to support the hypothesis that hemodynamics plays a large role in the creation of cardiac malformations, both mechanical and genetic. So shown here are just some classic animal models. These on the left are all done in chick for the mechanical, and on the right, these are done in mice. But all of these are changing subsequent hemodynamic circulation, and basically these are formed before the heart is four-chambered, and they create all these defects when the heart is four-chambered. And so one of these, I'm just going to point out a couple, is this left atrial ligation. So the suture is passed over the left atria, and it's pulled tightly enough to arrest flow. And that will recreate the single ventricle defect that we just saw on the previous slide. And you can actually you can reverse that defect. In the chick, it's just a day later because of its rapid development, by doing a clip on the right atria, and then you'll have a four-chamber circulation again. And then over here for our genetic perturbations, this is just showing one PIX2 mutant. So this PIX2 mutant is basically the outflow tract here is not it's not adapting its regular spiral structure, and instead it's linear. And so this causes a number of flow changes throughout development, and it leads to a bunch of subsequent large cardiovascular malformations. And this at the bottom is a deletion in the BMP1 receptor of cardiac neuroquest cells. And this leads to persistent truncus arteriosus, or what should be the aorta and the pulmonary artery are not actually dividing, but they remain one vessel. As an engineer, our goal is, especially as a bioengineer, to engineer new regenerative strategies. And so we're really thinking of how can we improve this? How can we take this apart? How does it function? And what can we do? And so in order to address problems at their root, we first have to understand how they're formed. And that's why we focus on cardiac development. And so going back to all these outflow tract malformations, we can go back to this critical stage of development. So this is an early looping phase of the heart, where you have a single ventricle, single atria, then a single outflow tract, and then up here at the top, these are the frontal arch arteries, which will form the great vessels. And so our overall goal is to elucidate the developmental basis of abnormal cardiac function to potentiate regenerative medical therapies. And so how does the heart form? Well, it begins with the specification of myocardial and cardial endothelial cell precursors during gastrulation. Then these fuse, the cardiac fields fuse to form the cardiac crescent. And then you have an inner endothelial tube. Sorry, my headphone is falling out. And then an outer muscular layer. So you have the single heart tube phase. Then the heart will loop once to the C shape, it'll loop again, S shape, and then it'll septate or divide into 4 chambers. And so here you can see basically the difference between the human timeline, and then below is the chick, since that's the animal model we're focusing on here. And you can see the chick develops so rapidly. So in order for this to be 4-chambered, it's actually 4-chambered by day 7 on the chick, which is HH31 for the staging that we use, Hamburger-Hamilton staging. By day 10, the valves have finished elongating and they're in their mature form. And so that's accomplished by day 50 in the human. And you can see down at the bottom, the volume changes are actually accurate from like 24 hours, one change to another in the chick. And what you can see from the nano CT scans are how the frontal arch arteries actually evolve and develop. And so during development, there are 6 pairs of these frontal arch arteries, which will sequentially emerge, remodel, and then disappear. And eventually they'll form the great vessels. So they'll form the aortic arch and all of its derivatives. And here in this paper, we're going to focus on 3 different stages. So these are early in development. One is at HH18. And so that's day 3, and then HH24, that's day 4, and HH25, that's day 5 of development in the chick. So all of these are before the heart has septated into 4 chambers, but these are really critical stages of cardiac development where a number of events have to line up in order for proper cardiac formation to continue. And so of note from the HH18 to the HH24 is that you have this arch artery 2, this 2 pair is regressing, and then the 6 grows in for HH24, and this 3, 4, 6 pair, this is what will mature into the great vessels. So this 2 will disappear, that'll regress, your 6 grows in, and these are the mature pairs, and so they still have a lot of more morphing to go through before they actually form the great vessels. Those are the stages we'll focus on. So all of this growth is accompanied by changing hemodynamic function. So here we have all the stages, the days. So we're focusing on 3, 4, and 5, and you can see how the heart rate and the systolic and diastolic volume change for all of those stages. And then an important emphasis of this study is its cohort-based nature. And so often biology is studied in 2D, and it's studied with the goal of determining the mean or average response. But these complex geometries are 3D, and what you can observe from them in 3D versus 2D is very different. And there's a lot of noise in the data that is usually disregarded. So if you're only taking an average model, then you're missing out on basically the natural variation that exists for whatever stage you're looking at. This green model here is actually from a paper we did with a collaborator a few years before this cohort-based study. And they used an N of 24. So they had 24 for the right side, and they had 30 for the left side. This is with the Pecan group. And so basically, they would make sure they had at least 3 arches on the right and 3 arches on the left, and then they would just average all of these together and have one model, and they ran this one model. And so there was a lot of information that we were able to gain from those simulations, but then also a lot of details that now we have because of the cohort-based approach. So a lot of details that were also missed. So computational models provide a powerful tool for understanding and interrogating biological events. And shown here are whole organ and tissue scale. So this top one is for that left atrial ligation that will produce the single ventricle defect. And we can see both the control and the LEL. And basically through the computational simulation, we can see how the instantaneous flow change induced changes in wall shear stress, which is sensed by the endothelial cells and quickly translated into cellular molecular level changes, so mechanobiological changes. And so we could really map those out, and that all of that was captured with CFD, computational fluid dynamics. And then down at the bottom, this is a soft tissue model from the Tabor lab, actually. And here they're tracking tissue levels to give the displacement field at both, and actually HH5 and HH6. And so basically the embryo starts and it's this flat disc. And then so that's HH5, it's flat, and then it has this head fold formation, and that forms the first 3D movement for the embryo. And that actually is when the heart field forms, so initiates heart development and everything. And so this is something that the Tabor lab was looking at while I was still in there. And basically from the computational models, they're able to compute strain field and really understand what forces are necessary for this head fold formation. So this combined experimental computational approach is an essential part of how we understand form and function, or how we attempt to assess the full picture of what's going on. So there's only so many measurements that you can take experimentally. And then if you do proper computational simulations, a lot of times you can fill in the holes and you can get a lot of values that you're unable to obtain experimentally. And so this is kind of our approach in general for assessing cardiac function. So we go back and forth between computational modeling and then in vivo validation assessment, mostly via ultrasound. Maybe you're going to take some stress strain measurements, and then that helps us to develop predictive animal models of congenital heart defects. So our particular form of computational modeling for my group is computational fluid dynamics. So here, these are just live chick embryos that are gonna undergo some kind of defect. So you can see the arches here, everything that we'll like look at now from reconstructions. Here's the outflow tract and the heart. This is the atrium. And so in order to perform our CFD simulations, we need a flow curve. So that's something that's really important. And so we attain our flow curves from pulse wave Doppler. So we perform ultrasound, and then using the Doppler, we're able to get velocity. And then we have to make some assumption about the velocity profile, and we're able to convert this into flow curves, and we use that for input into our simulations. So shown here, these are for all of the outflow tracks for the 3 stages that we're looking at, HH18, HH24, and HH26. And then over here on the right, this is in all of the arch arteries. So we can see how it changes, and then there's just standard error bars along the curves. So you can see that there's around an N of somewhere between 8 and 14, or 8 and 15 for all of these curves included in those error bars. So for our modeling, we took the multiscale approach. We began with a 0D electric analog model. And basically, we searched literature to see what already existed for the chick embryo. And we found this Yoshigi model where they were measuring pressure and flow simultaneously at the aorta. And then they were fitting this to different types of models. So it was perfectly exactly what we were looking for. And so we took their model and then we adapted it. So first so we'd like to create a 0D model of this whole PAA system. And so this is all the output from the PAAs, but first we have to adapt it in order to have our 2 cranial outlets, which are up here at the top, and then our caudal outlet down at the bottom. And biologically, there has to be a 90-10 flow split, so we have to enforce that. And then we found that the inductor actually wasn't needed, so it wasn't necessary, so we got rid of the inductor. And then our next step was to add in basically a resistor for each of these arch arteries. So, there's 3 on each side. And so here you'll have those 6 arch arteries. And then this resistor here is basically what it has to go through, the vessel that it has to go through in order to get to the bounds. So the bounds will be outside, like you can't see them outside of this 3D reconstruction. So here, these dash boxes are our cranial, and caudal branches. And so this will be part of the dorsal aorta where flow exits here. And then the outflow tract itself has to be represented by a resistor as well. So the reason that we're representing all of these vessel parts by a resistor is because it captures flow dissipation, so pressure drop across the vessel. And these arch arteries are in a really stiff pharyngeal mesenchyme. So they're really rigid. They're not moving. We can see that when we do 2-photon microscopy on them, but they're also really primitive vessels at this point. They don't even have smooth muscle cells. So there's really primitive vessels and these are really stiff sheets. And so that's why we represent them all with resistors, versus a capacitor would allow your vessel to actually deform. So the capacitor is basically, if your vessel is straight, and then it deforms into, I wish I could draw on here, but it's basically the ability to store a little bit of flow on the side as your vessel deforms. So some flow will continue to pass through the center of the vessel and some will stay on the side for the deformation. And then an inductor would be inertial terms. First we did our 0D model, and then we did reconstructions for a 3D model. And so we sent our embryos to nano-CT, and from nano-CT, we were able to get around 2.1 to 3.5 micron voxel scans. So very high resolution scans that we're able to get for these chick embryos. And from here, we can actually just reconstruct our regions of interest. And so then we combine the two into this multiscale model. So multiscale because you have both 3D for your region of interest and then 0D for downstream parameters that are being represented. And that is just represented by Ohm's law. So we have V is equal to IR, and our voltage is pressure, of course, which is actually driving our current, which is flow. Okay, so this was the model that we originally created when we were looking at our vessels and trying to represent them all, and under the inception that, okay, these are rigid walls, we don't need any kind of deformation over the cardiac cycle. But then we also had to update this. So this was fine for the HH18, it was fine for the HH18 models, but for HH24, we had to add in another dissipation here for the aortic sac. So we basically added in another resistor here in order for the flow to go up through the sac to reach these 4 upper vessels. So to reach these 4 upper ones, there's this resistance here. So we just updated our model from that. And so that's how we came up with this high resolution imaging and flow measurement, multiscale numerical analysis pipeline. And so we have our Doppler, we have our 3D reconstructions, and then from our 3D reconstructions, we're going to get the center lines. We do that using the vascular modeling toolkit. And then along the center lines, we're going to take measurements at 10 equally spaced sections for each of our arches. And so we take that, and so that way we can get all of our basically all of our morphological data, and then we can also get all of our hemodynamic data once our simulations are performed. And then these are the simulations that we're running. So there are these 3D OD simulations, 3D 0D simulations, and we have to tune our bounds. So our bounds are those lump parameter models. We have to tune it for every single simulation, and we tune them by using the fully lump parameter models as well. So here we have for our HH1824, and then it's an even different geometry because it's septated for the HH26. These are lump parameter models, and we use these to tune for the 3D, and then later we also use just to characterize the vessels in general. This allows us to combine all the hemodynamic and morphological data. So already, just from studying the arch arteries in 3D, we were able to learn a lot about the shape of the arteries. So for instance, with the previous collaborator I think they're the first to do CFD models of these arch arteries. So they showed us a lot about basically like streamlines, how the flow is partitioned, some general wall stress and pressure. But one thing that they assumed was a circular cross-section. And here we're able to see that like each stage actually has a distinct shape, meaning that, so here for our day 3 or HH18, it's actually elongated from right to left. So this is right to left here, and then this is cranial caudal going up and down. So it's elongated from right to left for day 3. And then it becomes more circular, and it goes into a perfect circle at what we call HH26 slow or day 5 slow. And then it actually elongates in the cranial caudal direction, and it keeps elongating that way for HH26 fast. So I'll go more into those subtypes as well. And so we can measure these shape changes by using these ellipticity. So if it's 1, it's a perfect circle. If it's 0, it's complete ellipse. And so here are just average models for each of the stages. And then this is showing how it changed along those 10 sections for the subgroups that we found in day 5. And so since we had these 3D models, we're like, okay, let's take as many measurements as we can. And so some of the other measurements that we took were basically branch splitting angle. So we're like, oh, does the angle that the flow actually hits each of those arches, does that affect, and how does that change? We also took length measurements. So all of these measurements are along the center line. And for these branch splitting, basically, we dropped a perpendicular bisector to the center if you just bisected the inlet. And then wherever the center line of each arch hits this bisector, the perpendicular bisector, that would be the angle that we would take. So we didn't find any significant changes either for the angle or for the length. for these HH1824, we found, I think, a couple for day 5 or the HH26 ones, but we were just happy that we could actually have all this information and get all this information. And so the other thing that arose just from the scans were this subset. So there's actually a large change between HH24 and HH26. And we could see how that change was taking place. And so that's why we called some of these HH26 slow and HH26 fast, and then one that was just a regular HH26. So shown in gray is an just average model. So an average model from the 5 HH26 nano CTs that we have, we averaged them all using statistical shape modeling. And then this is just showing you like what the slow, the regular, and the fast look like compared to that. So and this peachy color is all the embryos that exist. So we just had one slow, one fast, and then 2 for the median. And all of these shape changes, of course, influence your velocity profiles, which will then influence your pressure and wall shear stress measurements, especially your wall shear stress measurements. So that's why it's important to capture and to really take those into account. And so here, it's just, you can see how the profile changes along the arch with the shape. So one thing that we found once we ran the CFD was we found that pressure increases dramatically across stages. So it increased 32% from day 3 to day 4. It doubled from day 4 to day 5. So you can just see the population-based models down here, and then the general trend up top. And then wall shear stress stayed at the same level. So the embryo, or the body in general, is a lot more sensitive to changes in wall shear stress, really small changes in wall shear stress, since those are sensed by your endothelial cells, can actually lead to big changes. So you'll see that there's some spikes or hot spots for these like red zones. So those are significant, but it did stay within the same scale. So it was a value that was important to maintain across these 3 stages. And then here's how the flow distributions for the arches, how they changed across the stages. So it was most significant with the 4th right. So the 4th right is what will form part of the aortic arch. It's the 4th right in avian model. It's the 4th left in mammalian model, but it's always the 4th arch. And then also in 3 left and the 4th left as well. And so these are definitely characteristics of the fact that 2 is regressing, 6 is growing in, and then 4th becomes this dominant arch. But it becomes dominant arch as in it takes up most of the flow, but that dominantness actually changes as the arch arteries continue to remodel. And then up top, you can see how the circulating blood volume really changes dramatically. So in black is the HH18, and blue is the HH24, and then for the HH26 or the day 5, it's almost double the blood volume is now circulating. So it's huge change taking place there. So these aren't drawn to scale, these here. And so we have all this hemodynamic data and all this geometrical data along the length of the arch. And we decided to put all this together and see what relationships we could quantify independent of location of the arch. And so we performed linear regressions. So here you can see wall shear stress versus area and diameter. So we have minimum diameter, maximum diameter, and area. And we have that both for HH18 to HH24, and then HH24 to HH26. And we found statistically significant trends in 14 cases, showing that wall shear stress is a significant driver of the changes taking place. And then we did the same thing for pulsatility, so max and min pressure over a cardiac cycle. And we found 7 cases of significant changes. And so this was a secondary driver to wall shear stress. And then with all this information, we're able to make these least variability and max variability charts. So basically, the whole reason that we're doing the cohort is because we want to know which regions are really likely to vary, which ones are really tightly regulated. So like, where does it matter that if we're seeing changes along the arch, and which parameters matter the most? And so here we can see, okay, so this is least variability. 1 will be near the outflow track, and 10 is going back over here towards the dorsal aorta. And so we can see, okay, pressure and diameter and wall shear stress and all of those for HH18 are significant and near the they're near the outflow track, versus for your HH24, like your 10th position is where you'll have more of this least variability, and then for your HH25, you don't have as many pressure and wall shear stress tens in one spot, so that kind of varies. So maybe you'll have, here we have the 6 right and the 6 left, where we have just a couple trends that are least variability zones. So this information we really wanted to use to help us know where to look on a cellular and molecular level. So what's most tightly controlled in development. And this is another way that we can look at it. So these are deviation from mean graphs, where you'll have some kind of hemodynamic variable, so principal wall shear stress, versus some kind of morphological variable, and each of the sections along the arch are all represented here, so you can see them. And then you can see, okay, this is varying more for the hemodynamic variable versus this is varying more for the morphological one, and this is more tightly controlled. And we can look at that for each of the arches. So just another way to look at least variability per region. And so whenever you do CFD, you definitely have to validate it as well. And one of the ways that we used to validate it was, so we only impose the inflow curve. And so then we use measurements from the arches and the dorsal aorta to just validate that what we also measured via ultrasound, that we could recapitulate that. And so we compared this against both the Yoshigi paper, which is where we got the lump parameter model from, and then what we calculated from ultrasound, and saw that they matched up fairly nicely. So with doing CFD, we're doing them at exactly a single time point. And so we also really wanted to be able to extrapolate data between the time points more. And so for this, we use statistical shape modeling. So I have some videos on the next page, which I'll show you. And then down here at the bottom, there's basically a summary from the resistors. So from our lump parameter models, we can also take all of those parameters, and it can help us just characterize the functionality of each of the arches. And we can see how that changes across stages as well. So we see that the most significant changes are 4th left and 6th right. So you can see some of those trends here. And then, so we took out the arches that were growing in and regressing when we're looking at HH18 to HH24. So here we can just look and see that there's a linear movement for this HH18 to HH24, trying to match this template of one stage to the next. So we're trying to, this is an average of all of the 5 like HH18 models, an average of the 5 HH24 models, and basically we're like, what displacement field is needed to morph one into the next? And then at the bottom, we're doing the same thing, but we're doing it for the much larger displacement from the HH24 to HH26. And so here we can see that there's like not just the linear movement, but so much torsion that has to take place. And there's a lot that's going on with the heart as well. So between HH24 and HH20, it's actually the outflow track is rotating and it's lengthening. So the arches are both lengthening and rotating 20 degrees. And all of that influences the shape changes that we're seeing as well. These resistance area plots are another way that you can really understand the functionality of the arches. And so here we can see that, okay, both the smallest disappearing ones, so to right and left for HH18, like these had the highest resistance, like that makes sense. And then we see that 4th left for day 3, similar to 6th left. Those are both growing in, and so they had a similar trend here. And then we can also see other trends, such as the increase of resistance of established arches. These are also being at a similar level. So these are just some of the information that we can take away from the lump parameter models. In addition to their tuning, we can really understand the functionality. And so we also obtained a single value for each arch. And we plotted those resistance area, Walsh stress surface area, flow volume to see if we could obtain other different trends for these. And so it's just showing just some simple trends we're able to obtain for that as well. And so basically from characterizing these control phenotypes, what we learned is the distinct shape changes from one stage or one day to the next, and that pressure and Walsh stress were major drivers of growth. And our 0D boundary conditions are what allowed us to have these pulsatile simulations and also to functionally characterize the network. And this is only possible because we were doing the subject-specific cohort-based and silical study. And so that can really help us to understand variance and tolerance for both hemodynamic and morphological factors. And so the overall goal of all of this was also to understand, okay, how does this defects arise likely with small deviations from normal. And so if you want to understand the small deviations from normal, you first have to know what normal is. And then we now have like arrow bars on what normal is. And so then after characterizing everything for all these control embryos, then we went back and we did some mechanical perturbations ourselves, and we're able to compare them against all of this data that we had from the control embryos. So this is not in the cohort-based paper, but this is a large reason why we did the cohort-based paper. So I just wanted to give you a preview of what this enabled us to do. And so since we have all these control embryos, then we could perform in silico perturbations, or we could do virtual surgeries on them. And then we could do those same surgeries experimentally, and then we could compare their responses. And then we could also understand more about the biological response and what's really going on with these mechanical perturbations. So for our mechanical perturbation, what we did was we did occlusions of just the flow. And so we're able to occlude flow within an arch artery of our choice by using nonlinear optics or 2-photon microscopy and femtosecond laser ablations. So the 2-photon microscopy is our imaging laser, which allows us to image deep in the heart without damaging anything. And the femtosecond pulses fit precisely in the 2-photon laser and gives us these really powerful brief flashes of light, which allowed us to do the occlusion. And so for the occlusion, the embryo doesn't have clotting factors early on, so we're not able to basically, a normal clot would form by disrupting the surface, and then you have fibrillin and this whole cascade to form a clot. So these occlusions are actually formed by lysing a bunch of cells, which become sticky and they stick together in a clump. And so basically what we do with the ablation laser is we grow a cavitation bubble, and we grow it until it completely blocks flow within a vessel. And then you can actually see the cells line up behind the cavitation bubble, and you lyse these cells, and that's when they become sticky and stick together. And so we did this, and we see all these diameter changes shortly after we do the occlusion, and we just carry this out for 24 hours. You can see these diameter changes. And we did this for both full and partial occlusions. And partial occlusions weren't done on purpose. We were trying to do full occlusions every time, but the embryo tries really hard to maintain this connection. So the heart's down here. So it tries to maintain a connection between the heart and the dorsal aorta. So So we're able to compare these experiments, the conclusion experiments, with our in silico perturbations, and basically, sorry, so for the in silico perturbations, we did both full and partial occlusions, but the embryo is not able to remodel. So we can see the instantaneous changes in flow, and then we're able to compare this to known adaptation phenomenon and basically propose some kind of growth model. So predict changes in morphology and compare this with actually happened. And so that's how we followed up on this cohort-based study. And just to show, the initial changes from those partial occlusions actually did propagate into a substantial malformation. So those arches are tiny that we were occluding. And basically here, this is a partially occluded embryo. And normally your aortic trunk should pass to the side of your pulmonary, so your pulmonary branch, and it should cross over and bend over. But here they stay parallel. And that is similar to transposition of the great arteries. So your aorta is not actually passing underneath, but it's staying parallel. And so this could be us seeing this forming. So with that, I'll thank both my PhD advisor, Jonathan Butcher, and our collaborator, Irene Vignon-Clementel. So all of this work was done during my PhD at Cornell in the Butcher Lab, and this is the Butcher Lab at that time. And I can take your questions.
Sorry, you couldn't hear our clapping.
Great. Yeah, so we do have some time for questions. Are there any questions for the speaker? Manuel, is that raising a hand physically? I don't understand these real world gestures in a virtual world.
These real world gestures.
Yeah. Okay.
Germany,
yes,
we do this. Yeah. I don't think you do that in the US. Just pointing at the ceiling. Well, first of all, a really great talk. I really appreciate you taking the time and being here and also sharing your science with us. Thank you for that. I'm always so impressed with the work that's being done on these small scales in these models. And we forget it, right? Because yeah, images are so large, but in reality, everything is small. Yeah. I was particularly fascinated by the very end because I'm interested in thrombosis and thrombolysis and etc. So how long do these bubbles exist? So do you basically, does the bubble occlude the flow and then you know, there's like literal physical stopping of the flow?
Yeah,
because
so we're actually
so short, right?
Yeah, so it's easy for so the flow is circulating, so it's easy for these like little gas bubbles that you're forming. So it's like the reaction being contained in this femtoliter size volume, and that's a cavitation bubble that we're seeing. It's easy for them to kind of get sucked away and just to go away, like to disappear. They don't stay for very long. And so that's why we keep focusing the ablation laser into the bubble to keep growing it. Oh, I see. Then, yeah, so we keep growing it. So it's easy, like you'll start a bunch of cavitation bubbles and then you're like, okay, like I need one to like grow and actually block the flow. So we're temporarily blocking it, but then that cavitation bubble itself will also like, it'll start shrinking, you know, it doesn't stay for very long. And so like why it's there, you're like, okay, I'm lysing all these cells, like why I see them piling up. And then the cells will also like make it harder because there's like less flow going through for the cavitation bubble to keep shrinking and like kind of drift away.
That's very cool. A quick follow-up question. So you said that there's basically, you know, no inherent ability for the blood to coagulate. Is this consistent among all species? Like we're initially forming without ability to coagulate? I've never thought.
Yeah, so that's actually why like, so the assumptions that we have for our models is like we have this, we have the rigid wall assumption, and then we also have a Newtonian fluid assumption. And the reason that we can do the Newtonian fluid assumption is because the cells can't aggregate. So they have nucleated and non-aggregated rigid blood cells at this point. And so it is true across species. Up to a certain point in development. And so during this critical time in development, it's true. And so I guess one of our like theories is that, I mean, that maybe you have some kind of genetic predisposition that will actually allow your cells to either stick together or, you know, clot in some way, which will change your flow patterns like during development. So, yeah.
Cool. Really appreciate it. Thank you.
Emma, do you, well,
Yeah.
but maybe before Emma goes, are there any other questions from non-hosts? Just real quick. Okay, maybe not. Emma, you can go ahead.
Yeah, well, first of all, thank you so much for sharing this work, and it was really beautiful sort of data visualization as well. I'm sure that took a ton of effort. So yeah, really, really cool stuff.
Thank you.
I have a small question. It's just like a detail that I'm like personally curious about. You mentioned that you use statistical shape analysis at one point. I don't remember what slide it was on, but could you elaborate on that a little bit briefly?
Yeah, so let me just go back a little bit. I can go back here it's towards the end. Yeah, so we used a statistical shape modeling. So Irene Vignon-Clementel, she's from INRIA, so National Computing Lab in France, huge, and she's from like the Paris branch. So this was actually developed, the particular statistical shape modeling that we used is, this one was developed at INRIA. So it's just one of our colleagues who helped us use this and was like, oh, this is, you know, an available research. It's open source. But basically here, so there's a couple different ways that you can do it. You can parameterize your geometries and you can try to do some kind of matching up of your geometries, like all the points of your geometries, and keep them matched up. This particular one uses diffeomorphisms, and it really prides itself on not doing like point by point comparisons in order to match it up, but rather it's deforming the underlying space. And so this is just the most likely way that one shape can go to another. And so there's different calculations that you can do in order to actually, you know, so most common is you're actually going to do point by point for the surfaces. Parameterize that.
Yeah, thank you. Yeah, and so you mentioned that it was an open source software through INRIA.
Yeah, and it's called Deformatica.
Oh, okay, okay, very
Yeah.
cool. Awesome, thank you.
Any other questions, comments, Radon? If you're there, yeah, perfect, great.
Hi, that was a great talk. Thank you so much. So I actually have a question about the rigid wall assumption that you made in the 3D domain. So I noticed that for the lumped parameters, you did include arterial compliance through the capacitors, right? So was there a specific reason why you didn't include, say, deformable walls in the 3D domain? And how do you think that would affect your results?
Yeah, so the reason, too bad I didn't include like any videos of like, so we have all these like 2-photon videos because that's how we do, I mean, that's how we do all the laser experiments and everything. But like from the videos themselves, you can see that the walls, like, they're not deforming. Like they're not moving over the cardiac cycle, they're not deforming. But it was a pretty easy assumption to make also because these vessels don't have a large ability to do anything. So like at first they're just like primitive layers. And then like after septation occurs, so that's when the cardiac nodal crest cells will like form the tunica media around these arches, and then that will actually form smooth muscle cells later. And so then they'll have more of a capacity to actually, you know, like deform, like, and they're also like not confined into this, like, it's a really stiff mesenchyme that they're confined in. So that assumption is purely based off of their environment at these particular stages. But if we keep going up stages, then we definitely would have to do deformable for later stages. And then for using it for the arterial, that we definitely need to use for the rest of circulation. Definitely needs a capacitor in there to be accurate.
I see. Thank you.
I have a question, is maybe a naive question, and I think you might have even touched on this early on in the talk, but clearly here all of the analysis was done in the chick embryo. If you think about the sort of spectrum of biomedical research, the chick is not widely used. So I guess my question would be, are some of these developmental changes observed in mammalian cardiac development, like a mouse, for example?
Yeah, the reason that the chick it's very so the chick is like a classical animal model basically before like all of these like genetic like new inventions like took off, because the mouse is so great for creating lines and having your mouse lines and doing like all these genetic perturbations with them, but it's very difficult to create lines in like chicks. I think the only people who do it are like the Roslin Institute in the UK, and they have like a couple lines like a glow chick, and the reason that the chick is used for mechanical, so when people were really looking at morphological changes, is because it sits on top of the yolk sac and we have access to its entire vasculature. So for the like mammalian, like you have to pull the like embryo out of the mom, and then it has the hair that you're going to need to like shave off and see. So like the 2-photon is like great for imaging inside the heart, but the chick is still sitting on top of the yolk. So I don't think it's not possible to do this experiment in mammalian. So that's why it's done in chick. And then I actually get asked I get asked a lot like why the chick animal model, because the mouse is just so generally accepted as an animal model. And I mean, I love the chick animal model mostly from my undergrad days, and because it's so easy to work with, but also it resembles human development so nicely. So most of what we know about the mechanics of cardiac development, it's all coming from the chick. And then like they'll be able to confirm some different phenomenon in mice, but mostly like it's easier to do like OCT, like MRI, like something where you're like some static just looking at something like change, in order to like be able to like see these structures, instead of something more dynamic where you're also changing factors on the scale. And then so for the chick, for the heart in general, so 4-chambered, and generally like almost the same as human. And then for their valves, the human has like a tricuspid valve, and the chick has a muscular flap on one side and then a single leaflet on the other. And that's the biggest change between chick heart and mammalian heart. And then something that people don't know is that genetically, the chick is actually more similar to human than the mouse is. So the mouse has all these like non-coding sequences and everything. And the chick is actually genetically more similar. It's just that the chick is a lot harder to manipulate genetically. But then there's all these new technologies. A lot of them are coming from like the Broner lab at Caltech, which is basically a chick genetics lab. So you can do CRISPR-Cas9 in chick, for instance, but you have to do, so you can do these like gain and loss of function genetic experiments in chick, but you're doing it on like each individual chick embryo, like kind of thing. So yeah.
Interesting. Yeah, thanks for that perspective. I think it makes a lot of sense to study it this way. Like you said, the access to the vasculature is sort of unprecedented in certainly in mammalian species. You just can't do it. So I think your arguments make a lot of sense. I was just curious.
Yeah. I'm happy for the question. Thank you.
Good. All right, maybe if there's any last burning questions real quick. Otherwise, if not, I think we can thank the speaker again, and I'll do that by clapping. I'll thank everyone also for attending throughout the semester, and then we are in the early stages of planning for the spring, so we will certainly be in contact with everyone through the listserv then. So we hope to see you all in the new year.